Okay, so I'll just start off with trying to explain what the metabolic phenotype is. And it's really a way of describing how metabolism responds to environmental pressure. So here's a model of global warming, and the environment will always impose some pressure on plant or even animal populations. So these individual plants can respond to the environment by expressing what it's got in its genome. So they can express gene transcripts which encode proteins, and these proteins are involved in enzymatic reactions to make metabolites. And it's this metabolomics here that I'm interested in. So when you see the word omics, it's basically meaning trying to measure as many things as possible. So genomics, transcriptomics is trying to analyze all the gene transcripts, and the metabolomics is a way of trying to analyze, quantify and identify all the metabolites in your sample. So once we can start to identify what these metabolites are in response to the environment, we can start to assign some functionality to these metabolites and then function to proteins and the genes as well. So I'm interested in this sort of metabolomics and using it in the way to sort of assign this function to the metabolites in response to the environment. So why is this important in natural systems? A lot of this research is in crop science and medicine. What importance does it have in ecology and evolution? So as I've just said, plants always have sort of abiotic and biotic stresses, and they can respond to this by changing the way they grow or changing the way that they function. And of course these two things are related. And what happens over time? Plant populations spread, animals spread, and so on. And what you get is natural selection and adaptation. Okay, so we see a lot of variation in traits, a lot of variation in fitness and survival. And in any sort of given population, we see a mixture of these sort of fixed traits and sort of plastic and inducible traits. Okay. So this is a very sort of methods session. This is what I'm trying to sort of get across is how we measure these things in the field and in the lab. So there's two main ways to measure these things. One is sort of metabolic profiling, and this is where we try to identify and quantify all the metabolites in a system. And the other one is more sort of semi-quantitative, and this is the metabolic fingerprinting. So this gives us a general idea, don't necessarily confirm all the metabolite identities and the quantifications, but it gives us a general idea what's happening metabolically within the sample. Okay. We can do this in the field. This is up in Scotland. So what you want to do first of all is to stop metabolism as quickly as possible. So you've got your snapshot of the metabolic profile in your sample. This is sort of boiling methanol on a Trangia stove in Scotland. And then once you've got your sample sort of fixed, you need to extract the metabolites using a variety of solvents. Okay. And then to get your metabolic phenotype, we use the principle of different metabolites have different weights. Okay, so here we have a pot of sucrose and a pot of glucose, and I've just circled there the molecular weight of sucrose is 342 and the molecular weight of glucose is 180.2. So we take advantage of this by, if we inject this sort of green sludge into a mass spectrometer, it's a big balance, a big pair of scales essentially, what we should get is a metabolic fingerprint like this. So on the x-axis we have sort of a proxy for molecular weight and intensity on the y-axis here. So on the top sample we've got an Arabidopsis petraea plant from Wales and another one from Sweden. And I've just highlighted in pink here areas where there's differences in the presence and absence of different molecular weights and differences in the intensity. So what we can start to do is build up a picture of the metabolic fingerprint of different species and different populations and so on. So of course we can't actually, once we get thousands of these metabolic fingerprints, it's impossible to tell by eye where the differences are. This is where multivariate statistics come into eye. So we get lots of different molecular weights, lots of different intensities. You potentially have tens, hundreds, thousands of samples you need to analyze. And I can't sort of say, oh yes, the major difference is this cell here. We need a way of quantifying these differences. And usually in the omics world, and especially in metabolomics, the sort of the multivariate stats of choice will be principal component analysis, that I'm sure a lot of you are familiar with. That's like an unsupervised method, and there's other supervised methods as well where you tell the program that there should be differences between samples, and it tries to find out what these differences are. Geneticists also like seeing the data in this sort of dendrogram, sort of hierarchical cluster analysis. So as well as a genetic tree, you can see a metabolic tree as well. I like using a program called SimCP, but I know Rob likes R and various other sort of programs, but it's up to you really. So over the next 20 minutes, I'm just going to go sort of around the world and sort of describe these metabolic fingerprinting techniques in 3 different ecotypes, or 3 different habitats. So one is an Arctic-Alpine, another one is a sand dune coastal system, and finally another one down here on the Antarctic Peninsula. Just to give you a flavour of the type of work we can do with metabolic fingerprinting. So this is Arabidopsis lyrata petrea. If you're using one of Bill's keys, it's a perennial, so you can skip that step. It's not an annual. And it sort of grows in sort of high altitude sort of places like this. This is Skye in Scotland. And it's sort of a bit of a post-glacial remnant. So when the Ice Age sort of retreated, these are sort of populations that survived in various locations around Europe. The Swedish population is quite young because the land is still rising, and those populations are only sort of a few thousand years old. Okay. So what we want to know is, this is some work I did in Sheffield, what are the post-genomic processes that are still present in these populations? Can we see genetic differences? And my role was looking at the metabolic differences. So using this principal component analysis and this metabolic fingerprinting, what we can see is very sort of nice distinct populations. So here we have one from Wales and Ireland. These are from Sweden. The blue is Norway. The red is Iceland. So between countries, we can start to see some nice sort of metabolic differences. But we have to be sort of hundreds of kilometers apart before we start to see these metabolic differences. If you're too close, this metabolic fingerprinting can't distinguish populations. So when you build up the PCA models, okay, we can work out that there's metabolic fingerprints that are specific for different populations, but what are those metabolites that are causing the difference? So this is what we call a score loadings plot, and these numbers are the molecular weights that are causing those differences. So if you do some bioinformatics, you can start to work out what are the likely metabolites that those masses belong to, and then you can start to overlay metabolite names and the country. So for example, down here you've got Wales, Ireland, and so on, these glucosinolates associated with those countries. Other glucosinolates, other metabolites are associated with pulling apart other countries. Okay. And then we can start to get a bit of a picture. So we know that those glucosinolates, these are very sort of sulfurous metabolites. If you have something like sort of bitter crest and sort of things like that, they're sort of very sort of sulfury tasting metabolites. And populations where you get differences in levels of herbivory, some have got the more sort of toxic, more sulfur groups on them. So you can actually use this fingerprinting metabolite data to explain some of the levels of herbivory in other parts of the study. When we look at the data like this, it's the same data set but explored in a different way. Again, you get these sort of nice clustering events, and then we can overlay other environmental information. So some of these clades, again, this Swedish clade here, based on its metabolic fingerprint, is a bit of an outgroup because we know that it's a much younger population. And we can overlay things like the coldest nighttime summer temperature seems to explain a lot of this pattern that we see in the metabolic fingerprinting. Okay. So you can use this sort of fingerprinting to build on a hypothesis, to make hypotheses for other parts of the study. And then the take-home message from here really is that here's a population from Norway, and then if you gradually cold stress and cold acclimate these plants, you see very sort of 3 distinct patterns of its metabolism changing as they go through this cold acclimation period. But if you have something like Ireland, their metabolic fingerprint looks pretty similar under controlled temperatures, under cold temperatures, and even colder temperatures. So in a way, the Irish populations are metabolically primed to deal with colder temperatures compared to something like the Norwegian population, which is quite a surprise. What else can we do with this information? This is like a bioinformatics view of metabolism in cells. So these are all metabolic pathways, and you can upload your metabolic information. If you have gene transcript information, proteomic information as well, you can start to build up these metabolic maps. And if it's red, it's been upregulated, or if it's blue, it's been downregulated. Or if it's red, it might be more prevalent in one population, and blue it might be less prevalent in another population. So depending on how you label it. But this gives you quite a nice sort of an overview of what's happening in metabolism as a whole, and you can then sort of start to target maybe pathways of interest and much more sort of specific information you can get from certain pathways to build on new studies. So that's what happens in the lab. When we actually look at samples in the field, the only way to prove that some of these metabolic pathways are as a result of local adaptation is to do reciprocal transplant studies in the field. So what you have to do is take maybe plants from Sweden, grow them in a Welsh environment, and take the Welsh plants, grow them in a Swedish environment, and so on. And you keep reciprocally transplanting all these populations around Europe. It's quite difficult to do, and the initial data looks quite exciting in that what we see, and in a way it's almost expected, is the metabolome looks like the environment where it's growing. Okay, so this is roughly 5.4% of the metabolome of the variation is related to the environment, location of growth. So that's saying that a Welsh plant growing in a Swedish environment looks like a Swedish plant, and so on. Okay, so the environmental signal is generally much greater than the genetic background. The genetic origin of the plants, the variation of the metabolome is less so compared to the environmental variation. So, but these studies are quite difficult to do, so hopefully we're going to repeat some of those. So sand dune coastal plants, we'll move slightly further south. This is a project, this is Rachel George's PhD. So she's not here today, she's hopefully handing in her PhD thesis today. So she's looking at these coastal species around locations across Europe and trying to find whether the metabolic fingerprint or the plasticity of this fingerprint is related to how well a species can respond to climate change and sort of warming events across Europe. So, a very dominant, fast-growing species, does it have a more variable plastic metabolome versus a species that doesn't respond so well to climate change? So these are some of the sites that Rachel's been sampling up, and quite nicely as well compared, so similar to what we saw with the Arabidopsis lyrata work, you get these nice metabolic trees. So she did the metabolic extractions, did the metabolic phenotyping, and so the first question is, can we actually see species differences between the metabolic fingerprinting? And hopefully it was a yes, and we can see this in her sort of metabolic fingerprinting work here. So that's quite nice. And the other message is, within some of these groups, within some of these species, you can actually start to see a population differentiation. So some of these might be associated with plants growing in the UK, some of them in France, and so on. So it's great having the tree. What's causing these differences between these samples? And again, so you can do some data mining, work out which molecular weights are causing these separations, and by use of, so this phlegm, I won't explain it in detail, but basically you can see that if you look for the loading scores of molecular weight 184, it's much greater in this species, and a similar trend in this other one. So different species will have metabolites that are really pulling them apart. Again, this is just a screenshot of something called metaboanalyst from Rachel's thesis. So you can see that some of these metabolic pathways, we need to go into it. This is a geraniol, so it's a very sort of volatile, sort of smelly compound, and in some species, that metabolic pathway is quite important. So when you look at a community, a plant community as a whole, you start to see that those species have been living next to each other for hundreds, thousands of years, why some species have evolved different pathways and others haven't. So within a community, it's quite interesting to see what the metabolic differences are between species to make up that community. I mean, similar with the reciprocal transplant work we saw with Arabidopsis lyrata. She also did it with some of the other sand dune species, and here we have Veronica. And the same message came across in the environment. The metabolic variation is associated more with the environment, not from the background of those seeds. So a British plant looks like a French plant when it's growing in France, and so on. So quickly moving a long way south now to Antarctica, on this peninsula here. This is the British Antarctic territory down here on this Antarctic peninsula. And so this is work that I did in January and February this year, so these results are quite new. And it's a bit of a pilot study, so we want to know in such a pristine environment what the metabolic plasticity and adaptation, what role that has in a new species or in a species colonizing new territory where you have a very low level of biotic competition and inputs. So what are the traits involved in this? Because we've all been told to innovate and sort of innovate our methods. We also looked at polar algae as well. So with polar algae down there, their optimal growth temperatures are usually lower than 10 degrees C. Now this is quite important for things like biomass and in algal species that are growing in the UK and in northwest Europe, in that during the winter months it's too cold to actually get decent amounts of biomass production in the UK for algal biomass. So if we can use maybe some of these polar species, we can actually increase productivity of algal biomass and high-value products in the UK. So with an algae, Cambridge University and the British Antarctic Survey, I just had to go down there and sample some of these species in situ. So it sent me all the way down to here. This is the southern tip of Chile, and this is the research base there at a place called Rothera. And so what we wanted to do is have a look at the algal and the plant sort of communities and the metabolic composition of those plants and algal species across these different sites. So these are different islands. The research base is slightly further north as well. And even though they're close together, this is what you can see. So this is Leone Island here. That's this one. Then we've got Lagoon is over here. And the photo is taken from Anchorage Island, which is where I'm on there. So even though they're very close together, they've got a very different geology. So some are volcanic, some are based on sort of very sort of hard granite, and so on. So this is some remote sensing data as well. So they can actually sort of do remote sensing of the underlying geology to say that yes, these three islands very close together, a very different geology, and you might expect a very different plant sort of communities on these different islands. So yeah, so they're very close together. So we might expect there to be some sort of differences in the metabolic composition of these species. And it's very obvious when you're actually there, you can start to see the differences in the soil type. So this Leone, it's quite a dark sort of foreboding place, and it's quite an interesting plant. You can't really see the redness on here, but the Lagoon Island is very red, and the Anchorage Island is very sort of smooth granite type rocks as well. There goes my algae selfie. This is snow algae, so it's called Chlamydomonas nivalis, and it's one species, but it goes through a red phase and a green phase, and you see this sort of blood red patches on the snow. And they've got a very active green phase where they sort of swim around and mate. Then they overwinter in this red phase where there's a big hard cyst, and they'll sort of melt, go into the rock, and they can survive for tens of years under this red phase. So I was studying this sort of terrestrial snow algae. Here you can see the green phase mating. So they're very active under the microscope. You actually get male and females, and they'll swim around and they'll mate like that. But they don't survive long under the microscope because it's obviously too hot, so you have to sort of catch them mating whilst they're still cold. And then this is the red phase is here. So once they've mated, they'll sort of go into this sort of dormant red phase. And this is packed full of a compound called astaxanthin, again, which is quite commercially interesting as well. And you can see a bit of a gloopy mess around some of the cells as well. So that's a way of protecting the organisms from a variety of stresses when they're in this dormant phase. So mosses, lichens, grasses and liverworts. So this is about as diverse as you get. In terms of the pictures, you've got some of the red and the orange lichens, and you've got the mosses here, Deschampsia, Antarctica, and so on there. This is a Steph Bockhorst, some of you might know. He's got some of his warming experiments down there as well to see what would happen to these communities if you warm them very slightly. So that's about as diverse as you get, and that's quite a green area for Antarctica. Of course, it used to be a different picture thousands of years ago. If you get your eye in, you actually can see this is a fossil fern, and you can actually see a lot of these fossils in some of the areas down there. So it used to be vegetated extremely well compared to what it is today. So for sampling, again, it's a bit of a different picture from sampling in somewhere like Norfolk and in Scotland. You have to go by boat. Those of you who've worked in skua populations before, you'll know why you've got a flag on your rucksack to stop the skuas from attacking you. You've got lots of tubes, and then you have liquid nitrogen, and you have to get them back to get them back to the research station as quickly as possible to analyse. You also get to wear fetching clothes like this when you sample out there, so no one cares because there's no one else around, so that's great. So what we can do, we're sort of limited to what we can do metabolically in the lab. It's a very well-equipped lab, but we can do some sort of scanning. Here you can see the differences in the pigment concentration of the green algae. Here we've got, this is a UV sort of visible wavelength scan, so you've got absorbance in the UV range here, blue, green and red. And then to the green algal species have lots of chlorophyll, but when you extract the red algae, you see this absorbance here. And absorbance is this astaxanthin pigment here. So it's just to confirm what we're seeing visually. I suppose it looks red, and these metabolic screens actually proves that it's red. Okay. So what's happening between the islands? So this is just one, again, this is total chlorophyll. So between the different islands, you can see that there are concentration differences between the islands. The standard deviation is extremely large. Maybe we didn't sample enough, but you can actually see when you actually sample them, there's huge variation in the concentrations there. So there are location differences, but within each location, there's a lot of variation. But that's something to consider when we hopefully go back and do a much larger study. So the other metabolomics technique we can use is something called FTIR, and then this, again, so rather than using sort of UV visible wavelengths, we can go much into the infrared wavelengths, and you basically get a bit of a fingerprint of different classes of metabolites here. So this is roughly speaking, you get polysaccharide peaks, protein peaks, lipid peaks here and lipid peaks there. Okay, slightly more complicated than that, but that's generally what we're trying to do. So us doing these FTIR scans of the algae species and the plant species out there, and you can see, this is a red algae, this is a green algae, and you can see differences in lipid composition and the protein composition and some of these other peaks there between the species. So it's quite nice. So to conclude then, what we can, hopefully I'm trying to convince you over this past half an hour that these metabolic techniques can be quite a useful tool to look at sort of metabolic traits in line with other sort of traits we see in plant physiology. And these traits can sort of vary with distance, something like tens of kilometers apart. Maybe those distances will narrow when you're in much more extreme environments like Antarctica. Some of these changes are related to the environment, like we've seen with cold and herbivory. And of course, these implications for population persistence and community structure. And then the other sort of major conclusion really is, this metabolic variation is largely related to the environment that we see, and less so on a sort of on the genetic background of the plant. Okay, a huge number of people to thank in Cambridge, Sheffield, and with the British Antarctic Survey, and for methods as well. And thank you ever so much. I'll finish on a penguin picture. There we go.
Okay, so we've got one question from Matt that's come up on Twitter,
Okay.
which is, can you use metabolomics to diagnose ecosystem health?
Yes, there's a big research group at the University of Birmingham who're using sort of metabolic sort of fingerprinting and markers to determine sort of ecosystem health and ecosystem services. You can definitely see sort of pollution markers and so on. So yeah.
Okay.
Yeah.